So with Koch's postulates, it was developed by Robert Koch in 1884, and this is where it showed the first connection between a single isolated microorganism and a known disease. What these postulates have represent was the criteria that must be met before a disease can be positively linked with the pathogen. Koch's postulates there's these four Koch's postulates, the first of which is going to be the suspected pathogen must be found in every case of a disease and not be found in healthy individuals. The second one is the suspected pathogen can be isolated and grown in pure culture. The third one would be a healthy test subject infected with a suspected pathogen must develop the same signs and symptoms of disease as seen in postulate 1. And then the fourth one would be the pathogen must be re-isolated from the new host and must be identical to the pathogen from postulate 2. So using these postulates, how can we apply it to this one, to a potential disease-causing agent in a mouse? So first, the objective here is to determine whether a pathogen isolated from a diseased mouse can be purified and used to inoculate a healthy mouse and cause the same disease. Upon causing the same disease in the healthy mouse, the pathogen is re-isolated as the same pathogen that can cause the disease in the original diseased mouse. So if we're looking at the postulates as our sort of 1, 2, 3, 4 steps to connect the microbe that causes disease to the diseased animal, we would follow it in this way. So for the first postulate here, it says the suspected pathogen must be absent from all healthy mouse and present in diseased mouse. So you can see here that in this healthy organism, it looks like they collected red blood cells, and when you look at it in the microscope, all they see is red blood cells. When they collect sample from this diseased organism or this diseased mouse, they looked at it under a microscope, and it looks like it caused a suspected agent here, which was the one that's shown here in green. So for the second postulate, the suspected pathogen must be isolated from the diseased mouse and grown in pure culture. So then it looks like they would take this sample from the suspected agent or from this mouse, and then they add it or grow it in a pure culture. Looks like here they're using an agar plate, and we have in lab use a certain technique that helps us sort of get pure colonies or pure cultures, and that would be the quadrant streak. And once they have that, they show that they're able to grow this suspected pathogen from the disease organism on this agar plate. The third postulate is then taking the suspected pathogen, which is now in pure culture, and it must cause the same disease when inoculated into a healthy, susceptible mouse. So they take this agent, it looks like it's then injected into a healthy mouse. The healthy mouse falls ill, becomes the disease organism, so that would be the third postulate. And then the fourth postulate is that you should be able to collect sample from the diseased mouse and then re-isolate the suspected pathogen from this newly diseased mouse. So that's how you would apply these postulates to a disease-causing agent in a mouse. So my question from this point is to ask you this. So if we were here at the end and we are looking at this causative agent, which is now newly isolated from this diseased mouse, and now it's in pure culture. Using a microscope, what type of cells do you expect to see if you were to look at the suspected pathogen from this newly diseased mouse? So if we were to take these cells taken from this pure culture here and put it onto a slide and look at it in the microscope, what would you expect to see? What type of cells do you expect to see? So if you're thinking it should be something similar to this, the suspected agent, that's on track. But keep in mind, though, when we had done here at the end, this is in pure culture. So if we were to actually take the sample from this pure culture and look at it under the microscope, what we expect to see should be the suspected agent, which in this picture here is going to be green. So we should be able to see a pure culture of the suspected agent under the microscope. So the pure culture of the suspected agent. So that's what we would expect to see underneath the microscope, or when we're using a microscope. If you thought about this picture where there's red blood cells there, additionally, that is on track. But just be mindful that here at the end, this causative agent was isolated in pure culture on an agar plate. And this agar plate is going to be growing up that organism. And from the picture, it's going to be that green one.